reprogramming fully reverses aging on the epigenetic level of a single cell. And so the idea is then trying to capture that mechanism and apply it to many cells in vivo, of course, not allowing them to go to pluripotency, but hoping to get some rejuvenation using some epigenetic rejuvenation and functional rejuvenation and transcriptomic rejuvenation by using the same mechanism that was shown to fully rejuvenate cells in vitro.
So hello everyone, I'm Nina, this is Nicolas, and today we're going to interview Yuri Deigin. He's CEO of the company YouthBio, and he's been very active in the rejuvenation field for several years. So hello Yuri, how are you?
Hi, I'm good, thank you. Thanks for inviting me to the interview.
Of course. So first of all, as an introduction question, could you tell us a bit about your company, YouthBio? What do you do? What are the goals of the company?
Sure, yeah, happy to tell you about YouthBio. So we're building gene therapies that are based on reprogramming, partial reprogramming, which really took off in, I would say, like 2017. In 2016, there was this groundbreaking paper by Ocampo et al. That showed that you can use reprogramming safely in vivo if you do it for short durations, and that's what got known as partial reprogramming. And pretty much since then, I got very optimistic and very interested in trying to translate that approach into therapies, and been trying to do that ever since. So YouthBio is focused on in vivo therapies for humans, and in particular, right now we're focusing on the brain and looking at neurodegenerative diseases like Alzheimer's, for example, and others. And so we're trying to use partial reprogramming therapies in the context of these diseases, of course, to eventually have a systemic therapy that can be used for many different organs for systemic rejuvenation or systemic slowing down of aging that can prevent the onset of, if not all, that at least many age-related diseases. So that's what we're doing in the context of YouthBio.
And so I read on the website of YouthBio that you are working with the Yamanaka factors. Is that correct?
Yeah. In our kind of version 1 or first generation of gene therapies, we're just using Yamanaka factors, but eventually we plan on using tailored combinations of factors for particular cell types. But for now, we're using the canonical Yamanaka factors, yes.
And one thing that for several years already, there are several results by David Sinclair that he used the OSK factors and not the M factor. Do you think this is something that is also important to use only the OSK factors, or?
Yeah, I don't. I know David tries to make a point that c-Myc is somehow more of an oncogene than the other 3, but of course, all of the Yamanaka factors can be called oncogenes because, if they're overexpressed, then of course cells lose their identity, they go to pluripotency, and then they can redifferentiate and create teratomas. So basically, if you overexpress, and in David's experiments, if they overexpress just the 3 factors in their transgenic mouse models, they see the same issues with mice dying just after 3 days, if I remember the paper correctly. But the goal with partial reprogramming is never to let that happen, never to let the expression of the factors proceed so far that you can have cells actually losing their identity or irreversibly losing their identity, because there's some sort of like a gray zone where the like kind of transient, maybe not a loss, but a suppression of identity, which can then be recovered. Once you stop the expression of reprogramming factors, cells return to their original identity, cellular identity. And so we've seen many successful experiments with partial reprogramming with the 4 factors. Basically, if you limit the expression of the 4 factors to just a few days, and of course, it's also in a tissue-dependent manner that we've learned now with a recent paper from the Ocampo lab showing that if you avoid the liver and the intestine, you can express Yamanaka factors for up to 10 days fully, safely with 100% of mice surviving. Whereas if you don't, you only have like a 4-day window where you shouldn't be expressing them for more than 4 days of consecutively 4 days of expression. So yeah, I don't think it's absolutely necessary to exclude MYC. I think the 4 factors can also be used safely, and everything is highly dependent on cell types, as you know, has been shown by the Ocampo paper, that some cell types, like liver cells or intestinal cells, they're very permissive to reprogramming, so they're easily pushed over that kind of safe line of no return, or safe line of reprogramming, where just after 4 days of expression, those cells may not return to the original cell type, and they may cause problems even without pluripotency, even without oncogenicity. Just the loss of function of those organs can cause problems for the animals. And so this is actually kind of what's underlying our general approach of tissue specificity. We think that the future of partial reprogramming is tissue specific, and you need to find tailored combinations for essentially each cell type. Maybe maybe some cell types can share kind of the cocktail, depending on, like, the less permissive cells maybe can have one version of the cocktail. The more permissive cells need kind of a toned down version of the reprogramming cocktail. And so this is but kind of in the long-winded answer to your question, the underlying idea of making reprogramming safer, one idea is, yes, maybe you can exclude some factors, but I think the ultimate idea is you really need to have tailored sets of factors for specific cell types that will never, ever let the cell go too far, basically, in the reprogramming process.
And Yuri, you're a long-time defender of the partial cellular reprogramming technology, and you're trying, as you explained it, to make a specific treatment for specific tissues. But in 2020, there was an article that generates a lot of fuzz. It was a preprint by Harold Katcher and Steve Horvath, in which they were able to reduce the epigenetic age using the opposite strategy. Instead of very specific tissues, specific treatments, they made a whole treatment for all the tissues using the factors harvested from the whole blood of a pig. So this paper had many secrets that were revealed a few months ago, when it was revealed that it was pig's blood, that they isolated the extracellular vesicles. So my question is, and that's a point that Harold do, that if you make a specific, probably he could harvest, or you could, or anybody could harvest the specific factors. I don't know if they are only extracellular vesicles or Yamanaka factors or other factors, but specific factors for specific cells. But he says, well, but it's not better to take all of the factors to all of the tissues? So that's my question. If it's not better to take a holistic approach instead of specific.
I'm a huge believer in some sort of centralized control of aging, and I think we agree with this with Harold and many others, in terms of maybe there is a central role for the hypothalamus and the various hypothalamic pituitary axes that guide not only the development of our organism, which is an established fact, but also the aging of our organism. And so that implies that there would be some sort of a signaling mechanism that synchronizes different tissues. And obviously, there has to be some sort of synchronization of tissue development, because we know that, tissues develop in concert. Your different organs don't outpace or don't desynchronize. Your left arm doesn't outgrow your right arm when you're developing. And also, more importantly, maybe during sexual maturity, everything is like on a signal starts developing, like the adult version of the organism. So there has to be a synchronization mechanism. And so the search for this synchronization mechanism, I think, has been ongoing for some time. And I was very interested in the work of Dongchen Kai, where he was looking at the hypothalamic exosomes and hypothalamic signaling, and showed some proof of concept that potentially you can achieve systemic rejuvenation through the hypothalamic secretions and administering hypothalamic exosomes harvested from, I think, very young hypothalamic progenitors to mice. And so, yeah, obviously the work of Harold with his elixir or E5 and the idea that you can find this blood fraction, because of course, if you're looking for a synchronization mechanism, blood factors are probably the most logical way, or blood is the most logical way that will synchronize across tissues. For conceptually, I have like very strong belief or agreement that eventually we might be able to find such synchronization, and whether E5 is the first approximation of that, it's not like we made mice live 10 times longer with that fraction, which is, I think, the ultimate test, whether we found the actual synchronization mechanism. So from that standpoint, I guess in the absence of us finding that system, from what we're doing with partial reprogramming, and in general, partial reprogramming, I guess, is acting on like the downstream level within the cell, intracellular mechanisms of aging, because I think the signaling then changes the epigenetics of the cell, the gene expression pattern of the cell, and ultimately, of course, it's the cells that are responsible for all the functions of an organ. And it's the cells, if we can rejuvenate the underlying cells, if we can rejuvenate their gene expression or keep their gene expression at the youthful level, I think that means we can preserve their function at the youthful level. And of course, that translates to the next level of preserving the organismal function, or the functioning of the entire organism at a youthful level. And so that's why we're looking at kind of the next level of functioning of the organism at the epigenetic level, and we're trying to intervene using known mechanisms that have been conclusively shown to fully reverse aging. Like reprogramming fully reverses aging on the epigenetic level of a single cell. And so the idea is then trying to capture that mechanism and apply it to many cells in vivo, but of course not allowing them to go to pluripotency, but hoping to get some rejuvenation using some epigenetic rejuvenation and functional rejuvenation and transcriptomic rejuvenation by using the same mechanism that was shown to fully rejuvenate cells in vitro. This is a very kind of long-winded way of saying, I mean, I fully support the idea of trying to find the signaling mechanisms. And I think the preliminary results that Harold has demonstrated and Steve has kind of confirmed with his epigenetic clocks are inspiring, but I think we're still kind of a bit far off from the conclusive proof that these are the exact signaling molecules that we can harvest and apply and get, like full control over aging or full slowing down of aging that can prolong lifespan by, factors or orders of magnitude.
Yeah, one thing that you said a bit earlier is that your company, YouthBio, is developing a treatment for Alzheimer, right? Using your approach of partial cellular reprogramming. And one thing about Alzheimer is that there isn't like a consensus about the cause of Alzheimer. First, people talked about the beta amyloid plaques, and then the tau tangles, and then some people talk about the microglia, that is not, the problem is not the neurons, it's the microglia. So my question is, like, what approach, what are you trying to do with your therapy? You're trying to rejuvenate the neurons or the microglia or the whole tissue? I don't know. Could you explain a bit?
Yeah, no, you pretty much answered it. Yeah, in the absence of a concrete idea of what's causing Alzheimer's, as you're absolutely right, there's still no consensus on what exactly is causal in Alzheimer's, and there might be many factors, and people have focused on beta amyloid. But yeah, our hypothesis is, as you absolutely rightfully outlined, is just rejuvenating the underlying cells, underlying neurons, underlying astrocytes, and maybe some other cell types that we plan to target with partial reprogramming, bringing back their gene expression to a more youthful level with the hypothesis that can first of all alleviate or prevent the onset of the symptoms or prolong the duration until those symptoms start manifesting themselves. So if we're talking like an Alzheimer's mouse model, because with humans it's still, there's no like 100% causal factor. Basically, our therapeutic hypothesis is that if we're able to epigenetically and transcriptomically rejuvenate the underlying tissues of the brain, that we can prevent and maybe even reverse the Alzheimer's symptoms. And this is what the operating hypothesis that we're working under. And we actually have seen some promising results in mice, including the lowering of beta amyloid using our therapy, which is not targeting beta amyloid at all, but yet as one of the markers of potential onset of Alzheimer's, beta amyloid is used, and we have seen a reduction in the treated group versus the control group in our experiments.
For example, you are targeting therapy to the brain, but maybe you can rejuvenate the cells of the brain or other organ, like the kidney. But if you only target one organ, maybe the circulation blood, if this coordination signals of aging are in the whole blood, they will age the organ. So you make a specific therapy to a kidney, for example, and you rejuvenate the kidney, but if it's only the kidney, the whole body can make older the kidney again. So it's not better to make many organs at the same time, target many organs at the same time to avoid that.
Yeah, like if you target one organ, will the kind of the aging factors make it age? I'm sure, yeah, that there is in general this kind of tug of war between kind of the aged organism or potentially pro-aging mechanisms in an aged organism and some rejuvenation that you're trying to achieve in a particular organ. Like, yeah, if you're trying to rejuvenate kidneys, for example, then those kidneys probably have kind of conflicting signals between your therapy and the rest of the body, and you definitely need to take that into account. But of course, if you're able to kind of overwhelm the pro-aging aspects of the body with your targeted therapy, maybe with a higher dose of the therapy, or maybe it's just more effective. Like if your genetic construct epigenetically brings back the pattern of gene expression in a particular organ to a more youthful state, and it's able to keep it there for prolonged periods of time. By the time the pro-aging factors can restore it to the baseline, maybe with the right periodicity, you're able to still keep the organ younger and just, I guess, younger is a loose term, but just more functional. But of course, our ultimate idea or hope, and one of the reasons why we started with targeting the brain, is that because we believe that hypothalamus plays a central role in aging, we're hoping that maybe by targeting just the hypothalamus with our partial reprogramming therapies, our rejuvenating therapies, that can lead to systemic effects. Like if we rejuvenate the hypothalamus, maybe it will start to produce a more rejuvenating kind of fraction or more rejuvenating factors that tell the body that it's actually younger than it is chronologically. And this is the hypothesis that we're still exploring and hope to explore in greater detail. But yeah, in the absence of that, we will need to target multiple organs at the same time. And that's absolutely the goal, long-term goal, that we might have therapies tailored to particular cell types, particular organs that will need to be applying at the same time, or maybe, separated by, weeks or months. But ultimately, yes, you will be rejuvenating a multitude of organs with the hope of that producing a systemic rejuvenation for the entire organism.
I have a question, because you are a real rejuvenation person. I mean, I know you really want rejuvenation. So you are now in a strategy of a private company with investments, but I know that in the past you tried like crowdfunding strategies or something like this. So do you think that in a private company with investments, you would be able to implement a successful strategy? Because when in a private company, you can't share too much because it's private. So you have to patent, and in other models, you could share, like for example, the crowdfunding model, you could share the results, the partial results, because the research is already paid for the crowdfunding, and that would enable a parallel research with many groups. Everything they discover, they share, and the other groups can catch that and stuff. So there are several strategies, and nowadays you are in this strategy of a private company. So I would like to explore a bit the pros and cons, the good things and the bad things of this strategy you are implementing now.
Sure, yeah, and it's a very deep question that I've thought about and struggled with for many years, because you're absolutely right. In the beginning, I was thinking about and tried a crowdfunding strategy, and at the end of the day, it's really like a little bit orthogonal things in terms of funding and IP protection. And you can do like open source science or open science, where you don't patent anything and keep everything open, or you can do the traditional IP protection route, which is regardless of the funding. You can do this. Academic labs which have grant funding, they also patent anything that potentially commercializable, and universities of course own those patents rather than the researchers themselves, and then universities then license them out to companies. Because at the end of the day, if you want to create a therapy, unfortunately, the reality is a therapy must have IP protection to receive funding from investors, VCs, and ultimately to be bought or in-licensed by a big pharma company that will then translate that into the clinic, into clinical trials. And without clinical trials, unfortunately, right now you can't have a therapy that will be used by people. So, and clinical trials are very expensive. So essentially you are a kind of a hostage of the current system, that if you want a therapy to be on the market eventually, it has to be in a way that a big pharma company will eventually be interested in it. And of course, for that, it needs to be patent protected. Now, the idea of this fully open research is being kind of popularized and tried by some kind of open science collectives that think that maybe we can do completely open science without any patent protection. But I think, at the end of the day, if you want to create a therapy, at some point you will need to have patents around it. And of course, what you do with those patents and how the eventual therapy that the big pharma brings to market, how it's priced, and maybe then it's open sourced, or maybe in some countries those patents will not be enforced. That's our secondary questions. But when you're actually building things from the ground up for this type of research to be translatable and potentially translatable to markets like the US, where it's really like big pharma companies that are putting therapies on the market here, it needs to have traditional patent protection. But yeah, in a context of YouthBio, we're also doing the traditional route. We actually just filed a patent for our results in Alzheimer's, in the mouse results in Alzheimer's, and we're proceeding with the traditional route. And in terms of the reason people want to keep science open, from what I understand, at least in the longevity field, is because they want these therapies, eventual therapies that will be created, to be accessible to everybody, to be, either very adequately priced or maybe even free for everybody. And I don't think patents are a barrier to that. I think you can still have patents, but eventually, once the therapy is available, on the market, you can have that therapy to be very cheap or free. Like, for example, I think vaccines are a good example. There's patents around vaccines, but once you're mass producing the vaccines, the per patient dose is very cheap and very accessible, like in $10, $20 range. So I think, to the argument that people make that, gene therapies are going to be so expensive, I don't think it's valid, because it's just a matter of scale. If you're producing gene therapies for billions of people, they won't be expensive. They'll be cheap because, scaling up, basically there's economies of scale that make things per unit much cheaper. So
okay, well, actually, this is all the questions that we had for you. So thank you so much for your time, for giving us this interview.
Yes, Yuri, thank you very much. We wish you all the success.
Yeah.
And well,
and hopefully the therapies from YouthBio become widely available for everyone and accessible as soon as possible, because rejuvenation is really imperative, like for us, at least us that are interested in this field, that see the gravity of aging and age-related diseases, right?
Yeah, absolutely. Yeah, my pleasure. And I definitely, share that. I think there maybe not enough sense of urgency among some people in the field. I think, we need to be working on creating therapies and bringing therapies to market, to patients, as quickly as possible. So thank you very much for the invitation. It was a great pleasure to chat with you guys, and hope to see you guys again.